Washington University School of Medicine in St. Louis receives a $9 million grant from the National Institutes of Health to study the life histories of breast and pancreatic cancers. It's part of the Human Tumor Atlas Network. The project is using advanced technologies to study individual cells. Researchers are creating 3D maps of tumors, analyzing how the maps change over time. Scientists are trying to gain a deeper understanding of cancer cells so that doctors can more effectively fight the disease. The Human Tumor Atlas Network is an effort to understand the lifespan of tumors.
So my role on the Tumor Atlas is to use imaging as a tool to map cancer. So you select representative part of tumors to indicate how they interact with each other, what type of cells are there. So that's why we need to know where we are selecting, the tissue samples we use, and imaging plays a key role in doing that.
Professor of Radiology Samuel Achillefu explains how from there they are creating three-dimensional maps of tumors and their surrounding microenvironments.
So that you see how cell to cell interact with each other in a three-dimensional matrix.
With 3D mapping, they hope to gain new insight by studying the disease progression and responses to treatments in up to 300 breast cancer patients and 300 pancreatic cancer patients.
We want to understand this in a three-dimensional fashion, meaning the map, the atlas. So the tumor cells, along with the immune cells surrounding the tumor and the stroma cells surrounding the tumor.
As principal investigator, Li Ding uses detailed genomic analysis to characterize the cells through single-cell sequencing. The ability to sequence single cells in a tumor lets researchers identify which portion of the tumor may survive initial therapy and continue to grow despite aggressive treatments.
And we will understand what's going on in every single cell.
To do this, they must first achieve single-cell suspension. They're using dissociative solutions to separate cells in order to sequence each one of the cells individually. This allows them to identify the immune cells as they are part of the map of the tumor's microenvironment. Color coding identifies the different cell types. They analyze how the maps change over time.
So we can bring them back together, knowing what are the genetic alterations in each cell and how they are positioned in relation to each other.
Allows you then to see how heterogeneous different types of cells that are present in this type of cancer.
Single-cell sequencing of the microenvironment tells scientists how the patient's immune system is responding to the tumor invasion. Ding says studying the disease progression will help them develop better treatment options for future cancer patients with similar cancer cells and similar microenvironments.
In the future, when there is a patient coming in with the same sort of genetic alteration, same sort of microenvironment, we can kind of predict if we apply for this type of drug to this patient, maybe this is going to be observed after treatment.
Achillefu says the information can help identify the best approach to get the immune cells to fight the cancer through immunotherapy.
There's no better treatment than asking your body to fight for itself. Okay, as opposed to giving drugs and letting the drugs do its job. We all have immune cells that are designed to fight against all kinds of invasion in our body. Cancer is one. And so it's a hot area because then you are empowering yourself to treat your disease of all types. And that's why everybody's excited because if properly activated, the immune cells, that's the best therapy you can ever have, because they know how not just to kill, but also to dispose of what's dead in the body. That's why everybody's excited about that.